Tebubio in partnership with PBL Assay Science. I am Afa Gazgarova, product manager at Tebubio, and I will have the pleasure of moderating this session from interferon and cytokines to innovation. As you know, accurate cytokine and interferon quantification is essential for meaningful biological interpretation, but it's also technically challenging due to a low abundance, subtype diversity, and matrix effect. Our speaker today, Dr. Alok Pandey, Vice President of Biomarker Translational Science at PBL Assay Science, will guide us through practical principles and real-world examples to help ensure assay reliability, reproducibility, and biological relevance. Dr. Pandey brings over 2 decades of expertise in biomarker strategy and high-sensitivity immunoassay development. His work focused on translating innovative assay technologies into fit-for-purpose biomarker solutions that bridge preclinical and clinical research. Dr. Pandey, thank you for being with us today. The floor is yours.
Welcome, everyone, and thank you for joining. I'm Alok Pandey from PBL Science. Interestingly, over the last several years, I have probably looked at more cytokine curves and strange-looking dose responses than I care to admit. And one thing has become very clear to me. If we do not measure cytokines, interferons, or any other biomarker correctly, we do not understand the biology correctly. So today I want to talk more about a practical way of thinking about cytokines and interferon assays. And we will also talk about some of the targets that we have, such as interferon alpha, interferon beta, IL-15, and IL-22. And I'll show you some real data where assay design, subtype coverage, and matrix tolerance completely change what you see in healthy donors, as well as some of the patients from various infections, and as well as patients who are on therapy. So this will be a very applied, data-driven session. So very briefly about who we are, PBL has been living in the interferon and cytokine space for more than 35 years, and we design and validate high-sensitivity immunoassays, recombinant proteins and cytokines, and neutralizing antibodies that many of you already use. And Tebubio is our European partner. They are very close to the local scientific community, as you all know probably, and they help with the access, logistics, and technical discussions. And the shared goal is simple. It is not about selling kits, but to make sure that data you generate actually reflect the underlying biology. And before I start my presentation, actually, just want to tell you why immunoassays are important, because anything that you're going to use for your precious samples, you need to be confident about that. And when you look at the sample, each sample is a person. And when you look at the sample tube, every tube is asking, why am I okay? So you should be able to answer that. And for that, you need to have very good biomarker strategy in place. So what we are going to cover here, so rather than going reagent by reagent, I want to build a framework. We will start with why cytokine measurement is hard, then move into specific case studies from interferons and some other cytokines such as IL-15 and IL-22. And along the way, we will look at subtype coverage, free versus complex cytokines, preclinical versus clinical matrices, and what good validation actually looks like. Everything I show today is real, pulled from our technical data sheets and based on our internal validation work. So why cytokines are hard to measure? And probably you have heard this oh, I'm not able to measure my cytokine, or whenever I'm trying to detect my biomarkers in various disease patient samples, we are not getting robust data. So the answer here is, in healthy donors especially, true baseline levels are often in the sub-picograms per ml range, meaning very, very low. Sometime during an infection or if patients are on therapy, you can see rapid spikes, and then it returns to the baseline actually. So on top of that, many cytokines do not exist in a single form. So they form complexes either with the receptors, or they form complexes with different kind of binding proteins. As well as if you're working with some of the metrics such as serum and plasma, they also have several binding proteins, they have proteases, they have heterophilic antibodies which can interfere with the assay readability. And if the assay is not carefully designed and validated, then you can completely miss real biology, or sometimes you can predict that the signal that you are seeing actually, those are false positive or the noise. So let's talk a little bit about the cytokine forms, what is free, what is complex, what is total, and we are going to cover this more into the later slides. But here, two examples we're going to discuss about is, number one is the IL-15, and the second one is IL-22. Physiologically, most IL-15 is not free. So it naturally exists bound to IL-15 receptor alpha. And this binding protects IL-15 from rapid degradation, it helps in extending its half-life, and also help with the IL-15 delivery to other cells in a highly potent form through a mechanism called transpresentation. So, and as well as free IL-15, like if you look at the free IL-15, that also exists, but that has very smaller and less stable pool. But most importantly, both free IL-15 and IL-15, which is bound to the IL-15 receptor alpha complex, both are biologically relevant. So the most meaningful measurement is total IL-15, which is exactly what our assay detects. Now, if we talk about the other cytokine, which is IL-22, that works the opposite way. So IL-22 always binds to the IL-22 binding protein, and it has very, very high affinity, but IL-22 binding protein neutralizes the IL-22, and hence like there will be no signaling from IL-22. So the IL-22 and IL-22 binding protein complex is practically inactive. And what our IL-22 assay is designed actually, that can measure free IL-22. So this is, and that is the biologically active form. And we will cover that in the later slides. So here the distinction, IL-15 receptor alpha enhances IL-15 activity versus IL-22 binding protein neutralizing IL-22. That self-explains why the two assays must be designed differently. And let's talk about some of the subtype diversity. Subtype, like some of the interferons, for example, let's go to, we will take an example here for the interferon alpha. It is a very good example for subtype complexity. So when you look at the human interferon alpha, it has 12 subtypes, a family of related proteins, and they have more than 85% sequence homology, but they are with distinct biological properties and receptor kinetics. So in vivo responses often reflect the integrated effect of multiple subtypes. Now, before any clinical work, oftentimes you have to start with your preclinical study. So let's take an example of mice. When you look at the mice, in mice, interferon alpha is also expressed as multiple subtypes. So preclinical models are just as complex like your clinical model. So this is why interferon alpha assay that only sees one or two or maybe three subtypes is not sufficient. You want something that reflects the global subtype ensemble. On top of that, serum and plasma, as we discussed in the previous slide, that serum and plasma have binding and interfering components. So the matrix validation is not optional. If someone is going to say, hey, we can just simply use the assay without validating in your desired matrix, probably that's not a good idea. So before you commit your samples for any new assay, it's always good to go for the full validation, so you know how assay is working with your desires, with the sample you are targeting. So some of the matrix validation parameter that one can use is the linearity upon dilution, parallelism versus the standard curve, whether they are parallel enough, and the spike recovery in real matrices. That will be able to give you more confidence in the assay that you are using. Now we are going to start with 3 case studies. So number one, we will begin with is the type 1 interferons. And with that context, I want to show you concrete examples from our type 1 interferon assays. So here we will begin with the human interferon alpha and human interferon beta. And then we will discuss about how they look in the healthy donors, different kind of infections, for example, influenza, and some patients with the multiple sclerosis, MS patients, and patients who are on therapy of interferon beta and who are not on therapy. So what kind of levels we are expecting. And then we will move to mouse interferon alpha data that illustrates why preclinical model requires subtype broad assay and why pre-screening is important. So let's start with the human interferon alpha, all subtype ELISA. So PBL ELISA can measure all 12 subtypes. This is the representation of all the 12 subtypes in the standard curve format. You can see PBL ELISA basically can detect all the subtypes. So the human interferon alpha ELISA that we are using here, the calibrator is actually international reference standard, which allows you to compare across studies and labs. And the assay is validated in serum, several plasma types, and tissue culture media. We have done a lot of precision work, the full extensive validation we have completed, and based on that, we can tell you the inter-assay CVs are on the very low side, which is essential actually for your work, which especially with the interferon alpha, where most of the readings are lying around the lower limit of quantitation of the assay. And the antibody pair that we have selected here that can recognize all interferon alpha subtypes. And this is important because biologically, you rarely see a single subtype. You see an ensemble of interferon alpha forms induced by infection or therapy. So a subtype restricted assay might dramatically underestimate that a total interferon alpha activity, because there are several assays available which can measure only interferon alpha 2A or interferon alpha 2B. That may not be able to give you the proper idea about the levels of the interferon alpha in the samples. So little bit more data about the validation. So what we have here is, you can see the matrix validation work we have completed in serum and plasma. So here we did the dilution linearity experiment. So we serially diluted spike samples, and then we measured the concentrations. And it turned out that concentration stayed between 80 to 120% of the expected value. And this means you can use this assay in the real clinical projects where sample may come as serum or different plasma types without having to redesign your entire workflow. And this is very important because another thing is, if you are able to demonstrate dilution linearity and you are dealing with low volume samples, so you can dilute your samples at higher dilutions, which have been validated already, and you can get, and if you get the data, you will have full confidence with that data. The concentration that you are going to get from that, those are derived accurately. Now we are going to show you some of the data for the healthy donors versus influenza patients. As I stated that in healthy donors, interferon alpha levels are very, very low, sometimes very close to LLOQ. So what we have tested here, close to 64 healthy donor sera we tested. Most of them were below the detection limit, but some of the influenza samples that we tested, out of 38 samples we tested, 70% were with detectable interferon alpha, mean they were above the limit of detection. So this is exactly what you would hope for, because a clean separation between true negative healthy donors and patients with active viral infection, this is a very good representation of that. So if the assay were less sensitive or poorly validated, you might see there is no difference between healthy and the infectious samples, and which would be a technical artifact, not a biological truth. And now we will move to the interferon beta, actually. So interferon beta is another key type 1 interferon with different expression patterns. The interferon alpha beta assay ELISA that we have, that has it has a wide dynamic range and is validated in serum and multiple plasma formats. Precision is again very tight in this assay. When we perform the spike recovery experiments, that also showed that the assay behaves well in real matrices. And this allows you to use interferon beta as a pharmacodynamic marker in clinical trials or various therapies, or to study its induction in viral infection or autoimmune conditions. Here is very good slide. I mean, the biological picture is very intuitive here. Here you will see the healthy donors have interferon beta levels at or below the LLOQ when we tested them, but influenza induces interferon beta. So those samples show higher levels, and you can see here in this slide with the influenza patient have high enough interferon beta levels. And some of the multiple sclerosis patients who are receiving interferon beta therapy, we saw robust quantifiable interferon beta in circulation for them as well. So this is what you want from an assay. It should map clearly onto biology and the clinical context. Good analytical performance, again, as I said again, and I'm saying it over and over again, like very, very important, but not sufficient. So you also want the readout to make sense in real disease and treatment scenarios. So it's not like you can just look at your samples in the healthy donors. You really need to check them in the various disease indications as well. Now we will move to the preclinical model. So let's talk about the mouse interferon alpha. So when we measured mouse interferon alpha in untreated baseline animals, we observed quantifiable endogenous interferon alpha in a significant fraction of samples across different strains such as CD1, BALBC, C57BL6. And this is important for preclinical study design. If you assume that interferon alpha is always 0 in untreated baseline animals, you may misinterpret your stimulation or treatment effects. So our recommendation based on these data is to pre-screen serum samples to understand baseline interferon alpha in your specific strain and housing conditions, which is important. And I think I just skipped one, yeah. And here is mouse interferon alpha endogenous level in different mouse strains. We have checked that. Now I'll go to the next slide. So this is the case study, IL-15 free and complexed. So now we will move to the cytokines. So next, so here is IL-15, which is a very, very powerful cytokine in both autoimmunity and oncology. It is also a great illustration of why assay designs matters, measuring only free IL-15 versus total IL-15, which is a complex of free and the 15 receptor alpha. And that gives you a very different picture of the sample, same sample. So we will talk about that in the next slide. So here is the crystal structure that was published. We have the reference here for the IL-15. But if you look at the IL-15, it has one of the most interesting and unique signal architectures in cytokine biology. So in vivo, IL-15 rarely acts as a free cytokine. Instead, it is primarily produced in a pre-associated form bound to IL-15 receptor alpha. So this IL-15 and 15 receptor alpha complex can be membrane bound or soluble. So the membrane bound version is used for transpresentation, very important, because in this transpresentation, one cell presents IL-15 to another cell's IL-15 receptor alpha complex. And this mechanism massively increases potency and shapes NK and CD8 positive T cell responses. Free IL-15 does exist actually, and we talked about this in earlier slides, but it is less stable and generally less potent than the 15 and 15 receptor alpha complex. So IL-15 is implicated in many autoimmune and inflammatory diseases because of its potent effects on cytotoxic and memory T cells. So accurate measurement of this target, meaning the IL-15, is essential. Now we will go to the design of the assay that we have, IL-15 assay design. And because this is a complex target and it exists in two forms, so the most biological relevant measurement is total IL-15. And our assay is specifically built to do this. So the calibrator is mammalian expressed, or you can say HEC expressed IL-15 receptor alpha complex heterodimer. And this means the assay recognizes both free IL-15 and IL-15 when it is bound to IL-15 receptor alpha, mirroring the real in vivo distribution. There are some ICH M10 guidelines there, and people are enforcing that we should be able to demonstrate parallelism in our samples. And in order to do that, actually, you need to have your standard, which is mimicking the native form of your target. And this is where, like, that's the one of the reasons we brought in this heterodimer, because that's the physiological form which is predominant in this for this target, and that's what we are using as a standard. I'll show you some of the parallelism data in the next slide. But most importantly, the assay that we built here that has sub-picogram kind of sensitivity, and this is one of the reasons we can measure endogenous IL-15 reliably. And in all the healthy donor serum samples or the plasma samples that we tested, and if you look at some of the other competitors, their assays struggle with this kind of measurement. They cannot measure endogenous levels. So the reason could be whether it is measuring just the free IL-15 and not able to detect the whole complex. So this gives you a much more complete and meaningful picture of IL-15 biology. Now we will look into some of the validation parameter that we had tested for this assay. So analytically, this assay behaves very, very well. Intra-assay CVs are around 2 to 4% only. When you look across days and batches, such as like precision parameters, such as inter-assay precision or intermediate precision, they remain roughly in the 10 to 13% range, and which is excellent for this concentration window. The minimum detectable dose, mean the LLOD, is approximately 0.15 picograms per ml for this assay, and the lower limit of quantitation is around 0.5 picograms per ml. We also performed spike recovery experiment in various matrices such as serum, plasma. We also tested tissue culture media because IL-15 is very relevant to CAR T, CAR NK kind of therapy. So a lot of people are interested in tissue culture media. So we tested that as well, validated our assay with these matrices, and it showed very good recovery within the usual 80 to 120% expectations. And this gives you full confidence that the assay performs across matrices that you actually use. So very important data. And I was talking about the parallelism. Almost everyone is talking nowadays about the parallelism. And here we are going to show you how the data looks. So in the first graph, you will see the parallelism data. This is like experiment shows that the diluted curves track the standard curve within the plus or minus 20% of the real value, which is a strong indicator that the sample matrix behaves similarly to the standard. And we also looked into the specificity. So basically, capture antibody block experiment was performed to look into the specificity. So when we pre-incubated samples with anti-IL-15 capture antibody, the measurable IL-15 signal collapses towards the LLOQ. So that's the beige one, beige color. And then isotype control does not produce this effect. So those are the blue bar in the bottom bar diagram that we have here. And so anisotype control does not produce this effect. So this is very clear that the assay is very specific to IL-15. And it's not like you are looking into the false positive or false negative data. This is interesting. If you now look at the comparison with the other ELISAs, we tested 3 different competitors who were well known and ran our IL-15 15 receptor alpha heterodimer on their kit, and they were not able to detect this target basically. So the explanation could be whether they were using either the free IL-15 only, or they were using the E. coli express protein, not the mammalian express protein. So competitor assay does not detect IL-15 very well, and this is one of the reasons many of the literatures that are out, people have talked about like IL-15 is very difficult to measure, but with the new assay that we have developed now, I think mindset is going to change now. People will start looking into IL-15 and they might be able to measure IL-15 more reliably compared to what they did earlier. So this is very, very important slide actually. So at some of the disease states, like, for example, when people were looking at and the patients who are under therapy, and you may falsely conclude that IL-15 does not change or does not exist. That was not the case probably. And now with the new build that we have, most likely we will be able to measure that. Now the case study third is IL-22 and free versus neutralized cytokines. This is another important target, and people always try to work with some of the complicated targets actually. So IL-22 is one of them. IL-22 is biologically active only when it is free. So the moment it binds IL-22 binding protein, it becomes completely neutralized and cannot signal. This is what we have demonstrated here at the mother axis IL-22 binding protein. You see the signals are gone actually. So that proves that our assay can measure free IL-22. So when we measure IL-22, the meaningful pool is the free and unbound IL-22 that can interact with the IL-22 receptor which is present on the epithelial and the stromal cells. So our IL-22 ELISA is designed to detect exactly that, free IL-22, because as I showed in this figure with the IL-22 binding protein experiment, it is blocking the signal. So this gives you a much clearer picture of IL-22 biology in inflammation and autoimmune diseases. Now we will talk a little bit more about IL-22. So IL-22 is produced by a range of lymphoid and myeloid cells, including Th17, Th22, Th1, RC3, NKT cells, and even neutrophils. So its main targets are epithelial and stromal cells, and it can be protective by promoting barrier integrity, but in chronic inflammation, it can also contribute to pathology. So IL-22 is elevated or dysregulated in several autoimmune diseases such as lupus, psoriasis, atopic dermatitis, rheumatoid arthritis, and it is often associated with IL-17-driven responses. So the PBL IL-22 ELISA that we have that uses a HEC express or the mammalian express IL-22 standard that detects free IL-22 and is validated in both serum and plasma samples. Some more performance metrics we have here. So let's start with some of the patients actually. So when we looked into the healthy donors actually, so we were able to measure all the healthy donors with our IL-22 ELISA kit. We looked into various different disease indications such as atopic dermatitis, psoriasis, rheumatoid arthritis, or the lupus patient samples. We were able to measure everything within the detection limits of the assay. When we are looking into the parallelism, actually, so this is the data that we have from the disease state atopic dermatitis patient samples. You can see that very nice parallelism we are able to see when we do the serial dilution of the samples. We also ran a standard curve in various metrics. We did with the standard diluent, we did in the healthy donor serum samples, as well as in the TCM curve pretty much overlapped. So basically it behaves very well in the standard diluent as well as in the matrix. But the most important is here, like when you, like the fourth graph, I'm not sure like whether you are able to see very clearly on the screen, but readability comparison is very important here actually. So the PBL ELISA detects IL-22 in essentially all the psoriasis, RA, SLE, and AD patient samples, as I indicated earlier. But some other competitor ELISA, and which is very popular actually, fails to detect IL-22 in a large fraction of those same samples. So again, why this is important, this is again indicating that assay design and the standard choice determine whether disease biology is visible or not. So based on what we have discussed, this is a practical summary slide. Whether you are looking at our assay or anyone else's assay, these are questions you should ask. First, what form does the assay detect, whether it is free, complex version, or the total? Second, which subtype or isoforms are recognized, which is important. We discussed more about the subtype, But at the same time, isoforms also matter. Like there are various targets where you have so many isoforms, so you really want to make sure that your assay is able to measure your desired isoform and you're not getting false positive signals. Sometimes in the isoforms you have a lot of homology and similar sequences. So target is very important. So you should be able to target your isoforms very well. Then only you can get some meaningful data. So those are some of the biomarker strategy you have to think about, you have to implement before you start even your preclinical work or the clinical work. And third is validation in your desired matrix. If you are purchasing a kit, kits are validated just for the serum samples, and you really want to use that for the plasma. My recommendation, you should still go for the full validation. Or let's say like if you are looking for a different matrix such as TCM or saliva, urine, or CSF, just don't go with the validation report that you have in hand based on the uh, validation part, which is done with the serum and plasma, because it may behave differently, or you will be surprised that it is not able to pick up signals in those metrics that you are interested in. So proper validation is very important. In order to do your validation, like during validation, if you have to tweak your assays a little bit in order to make it work for your samples, that's another thing you can try. But our recommendation is always go for the full validation. You would like to look into the sensitivity of the assay in your sample diluent as well as in the matrix. You would like to look into the precision of the assay, spike recovery, linearity, or the parallelism in real samples. And you should also look into healthy as well as in the disease state patient samples. And if the biology you see is completely at odds and what is known in the literature, that can be a red flag about either the assay or the sample handling. So since we are talking about all the assays, I just wanted to give a little bit company overview here. In the center, you will see our founder, Dr. Sidney Pesca, who is also known as father of interferons, based on his pioneer, a lot of work he has done in the interferon space. He was the one who in vitro purified in vitro for interferons actually for the very first time. And then he started his company actually almost 35 years ago. We are known for high quality products. Right now, we have 9,000 plus product citations. We have one facility in Piscataway, New Jersey. Amongst us, we, like all the scientists that we have in our company, we have close to 259 years of cumulative experience. We are preferred providers for the reagents as well as services to a lot of largest pharmaceutical companies as well as some of the mid-sized and the smaller biotech companies. We also work with the academic institutions. And we, as I think some of the products we discussed about, but we have some other reagents and the antibodies as well, actually. So known for the assay kits, especially within in the interferon space. Now we have R22 and R15 ELISA kits, which are very, very popular now. A lot of monoclonal antibodies, polyclonal antibodies, neutralizing antibodies. Those are some of the products we have. We have protein system. We can express proteins in E. coli expression system, as well as mammalian express system. Nowadays, when we are looking for the custom assay development, if you are working with any complicated biomarker or the target, and you're not able to find the right expression system, so you can rely on PBL, we can definitely come up with some solution for you. Our kits are known as Verikine and Verikine high sensitivity ELISA kits. Most of them are, as I discussed in the validation report, like many of them were validated using the real world samples actually. And our kits have been validated by many other CROs and some other companies as well. Effective reagents for specific research needs we have. For example, if you're working with cross-species universal type 1 interferon protein you're looking for, we have those proteins. We have neutralizing monoclonal antibodies for type 1, type 2, type 3 interferon receptors for signal knockdown. And we have also have the neutralizing antibody mixtures. So a lot of products we have. This is just a glimpse of some of the popular PBL type 1, 2, and 3 proteins, but we have many. As I indicated, more than 130 products we have, but these are some of the popular ones, and you can see the citations that we have. So sometimes you may be asking this question, hey, whether your antibody is going to work with my FACS work or whether your antibody is going to work with ISC. We may not have all the data, But the good thing is like people have used our product and a lot of citations are there. So based on that, we can always tell you that, hey, most likely it will work with your desired requirements. So obviously citations always help and it always builds the trust. So that's what we have here right now, more than 9,000 plus citations over the years. And this is interesting. Some of the analytes that we have, or the ELISA that we have, if you just compare that against Biotechne, which is a very popular brand, almost everybody knows R&D immunoassays. And when we compared against the Biotechne, sensitivity, specificity, everything was unmatched actually between PBL and the Biotechne. When it comes to the interferon alpha, we can measure all subtypes. Biotechne doesn't have that kind of capability. When we talk about the interferon beta, obviously our interferon beta levels, like the sensitivity is much higher compared to Biotechne. When it comes to the IL-22 or IL-15, they cannot measure some of the endogenous levels. We can measure, our sensitivity is better. We can measure free IL-22, which they cannot. We can measure total IL-15, which they cannot. So this is a fair comparison side by side. Just wanted to show you, because PBL is investing a lot whenever we are developing the products. So this is one of the reasons you can see that our quality is superior compared to our competitor. And your success is always our success. So we have full supply chain controls. We have validation ready kits, stringent QC controls all the time whenever we are working on any new lot. Before releasing, a lot of QC testing is done. And then we can always prepare the custom kits. Depending on your requirements, we can always return in the batches, and the bulk proteins are also available. So you can count on us. Some of the research area of interest we have highlighted, but nowadays in various other indications as well, you will see that the requirements of the kits or the reagents that we have, especially the interferons, are almost applicable to everything. So for researchers working in autoimmune or cancer, immuno-oncology, infectious diseases, inflammatory diseases, rare diseases, immunotoxicology, a lot of researchers are using our products. And how PBL and Tebubio support you is the next thing here. So to close, PBL's philosophy is always to design assays and reagents that truly reflect the underlying biology, whether that is broad interferon alpha subtype coverage, total IL-15 measurement, or free IL-22 detection in complex matrices. We also provide recombinant cytokines, neutralizing antibodies that you can use for mechanistic and pathway studies. Lot-to-lot control and thorough technical data are part of our standard workflow. Tebubio is our partner in Europe for access, logistics, and technical discussions. And with that, I'll stop here, and I'm very happy to take your questions and discuss specific applications you may be thinking about. Thank you.
In the QR box, if you haven't already, you will find it on the right side of your screen. And meanwhile, I lost the presentation. Sorry. Yeah. And while you're doing that, we will invite you to share your feedback with us. It only takes a minute, and it's extremely valuable to help us improve future sessions and select topics most relevant to your research. Thank you in advance for taking a moment to share your thoughts. Now let's take a look at the question you have submitted. The first question for Alok is, why do different assays sometimes give very different results for the same cytokine?
Oh, very, very interesting question, actually. And I think we covered this during our talk, how you are building your assay. And that was one of the examples, like for the IL-15, like if your assay is not designed to measure your right target, you may not be able to see the results that you are looking for. And ultimately, it would turn out that, okay, the result that you have with one kit versus the other kit might be very, very different. And so important context here is like you have to really understand your target and you have to design your assay for that target. especially like if that is like, as I said, like whether that exists in the free form or complex form, or whether you are looking for active form, then what that active form is actually. So you need to understand that, and accordingly you have to design your assays. At the same time, when you are looking for the isoforms, for example, and you have multiple isoforms which have very close proximity and they have almost similar sequences, so when you're going to target your protein, it may turn out that you are not only able to pick up your isoform that you were interested in addition to that, you might be able to capture some other isoforms, which is giving you some false data, false signals. And so the kit which is designed for that purpose versus the kit which is not specific for your target may have different interpretation and the different data. So this is why you are seeing different data from different assays.
Thank you very much. The second question is, what are the main reasons some assays fail to detect endogenous cytokines in disease samples?
So disease samples are very complex actually to work with. I talked about like some of the components that they have. For example, proteases are there, heterophilic antibodies are there that can interfere with your assays, especially when you're working with autoimmune sera where you have a lot of RA factors involved. So this is gonna impact your assays. Sometimes it may interfere with your readability as well. So important consideration is when you are building your assay, you need to uh, and you have to ensure that you are able to take care of those factors. You may have to work with some of the ingredients that might be able to knock down that effect, and then ultimately you will be able to measure your target. So this is one of the reasons, like some of the assays which are still talking about, like they have very high sensitivity, they are not able to detect levels in the disease state patient samples. And I'll go back to again, like because uh, I don't want to talk about some of the examples which we have not discussed, rather than going back to the same, like for example, IL-22, for example. If you're not able to measure free IL-22 and then you are looking for the atopic dermatitis patient samples, IL-22 levels are tend to be on the higher side in the atopic dermatitis or any other autoimmune sera samples. Now, if our free IL-22 you're not able to measure, then you might be thinking that, oh, in our disease state patient samples, IL-22 is not elevating actually, or it's not going up, which is not the case. So most important is the assay design and the validation.
Thanks, Alok, for your answer. Another question, why does it matter whether a cytokine assay measures the free form, the complex form, or the total form?
I think we covered this actually during our presentation. So again, when we talk about the IL-22, for example, like when IL-22 is present as a complex with the IL-22 binding protein, practically it is inactive. So you don't want to measure something which is inactive. You would rather like to focus on something which is active. You need to understand the biology. You need to see like which form is active. So based on the literature search, you may be able to see that, okay, or there's some of the structure you can study, and based on that you can determine like which target, which is your real target, where you have to target your antibodies for. So that is important. So, and at the same time, uh, with the IL-15 is a good example. Like it turned out that IL-15 is present as free as well as in the complex. So, but the total IL-15 is important because free is also important as well as the one which is bound to the IL-15 receptor alpha complex. So the total measurement is something you would like to look into. So this is where, like, rather than thinking about like which form or which particular isoforms are important, it's more important to understand the biology of your target, and then based on that you can design your work.
Thanks, Alok. And last question I see for the moment, why do interleukin 15 and interleukin 22 require such different assay designs?
Yeah, very interesting question. And as I indicated for the IL-15, because IL-15 most important is the total form, and total form is active because when IL-15 and IL-15 receptor alpha complex are together, they are active. As opposed to IL-22, when it is binding, it is forming a complex with IL-22 binding protein, it is practically inactive. So you really want to see only the free IL-22 for your accurate measurement, or to look into the activity of IL-22, you can only rely on the free IL-22. However, when it comes to the IL-15, you can rely on the total IL-15. So that's why the assay designs are different and practically opposite to each other. But it helps you to understand that why complex matters, and complex is very important for your like in order to make a final decision about your therapy outcomes.
Thanks a lot, Alok. for your answers and for sharing your expertise. And of course, thanks to everyone who took part in the discussion. Finally, if you would like to continue the conversation or discuss your own projects, feel free to reach out to our CRS team, support scientists across 4 care areas: RNA-based therapeutic discovery, in vitro modeling, biomarker mapping, and data analysis. You can contact us anytime, and we will be very happy to help you design or optimize your next project. Thank you again for joining us today. We hope to see you at the next webinar, and until then, have a great day.
Thank you all.